And so what are some of the risk points that you might see in the process of manufacturing an injectable, for example?
Well, I think when you start with manufacturing any product, the raw materials are pretty important. The grade at which you purchase and the source that they're purchased from are pretty important. So having a robust external audit team to look at material providers is pretty important. You don't want to purchase a lot of, let's say, vitamin E from a particularly dirty facility that no one's ever seen. And how would you know if it's dirty if no one's seen it? So certificates of compliance, trying to find other ways of leveraging assurances that the facility that materials are coming from are clean, and that the grade of the material is what it should be as well. So I'd say that's where everything starts.
Yeah, and I agree with that. So you have materials, and I think also when you're talking about aseptic processing, the people involved in the aseptic manufacturing process are typically, represent the biggest risk to contamination because, depending on the process, you have, facility controls, which provide sterile HEPA filtered air. You have rooms held under positive pressure, to, push the air out and keep airborne contaminants out of the room. But then you have people that are, inherent to the process, and they wear sterile gowns, hood to toe, where they don't have any, exposed skin. But still, even inside of a clean room with a person that's sterile gowned, that person still represents the biggest risk to the potential for contaminating a product during the manufacturing process, whether that be during formulation or whether that be during the actual filling process. And during the filling process is really, can be high risk because the product is temporarily exposed to the environment, right? And so you have during the manufacturing process, if when during that critical step, you have facility controls in place, you have your equipment operating. If your equipment malfunctions in any way and people have to get involved, then there's what's referred to as interventions during the aseptic process, and where people have to, perform more invasive interventions, and that always provides an inherent risk to the process and contamination. So you have to have mitigation steps. You have cleaning and disinfection. You have environmental monitoring. They monitor surfaces, they monitor airborne particulates, all as a way to detect different types of contamination, primarily attributed to the presence of personnel. And the presence of personnel inside of a clean room significantly drives up the risk of introducing particulate and microbial contamination to the environment. So I think James hit it spot on. And then, with material, you have materials, and then you have people that represent the two, probably some of the two biggest risks, maybe. To build on that, on the material side of it, you want to check your materials upon receipt, and, you can test them for bioburden to see what their bioburden levels are before they're used. You can also test bioburden of the formulation in process before it's rendered sterile through filtration, to see as a means to detect what the levels of bioburden are before you use the materials. And then the other piece I think is important with materials is the transfer of materials into your clean room. So a lot of the equipment and materials that you use inside of a clean room are typically sterilized, by either through depyrogenation oven or by autoclaving, using moist steam to sterilize the equipment, but some equipment you have to transfer into the clean room or materials that cannot be sterilized. And so you have to manually disinfect those materials. And I think that represents a common risk, not necessarily a high risk. In some case, it depends on the process, but I think transferring materials into your clean room is an important step that represents definitely a risk that's common to a lot of manufacturing processes.
I think one of the kind of counterintuitive things about aseptic manufacturing is, I think, kind of summed up in a portion of the USP, I believe it's chapter 70 for sterility testing. While the test is labeled sterility test, and people producing aseptic products, sterile products, need to perform a sterility test for release, the chapter itself says that a sterility test does not guarantee sterility. It is a sampling exercise. The only way that you can actually guarantee that a product is sterile is to filter and test literally every milliliter that you're producing, which is just not possible. So there's a huge burden that is put on a facility to look at all the processes, all the inputs, materials, personnel, equipment, flows, disinfection, all of the inputs for producing an aseptic product need to be monitored and demonstrated that you've got everything under control. And that is done through a microlab, microbiology laboratory. And they're really important for being able to go out and sample and validate, perform studies, and trend data to demonstrate that a facility is managing to keep their processes under control, because they can't do 100% sterility testing. It's always going to be looking at what are the chances, and those chances are summed up in sterility assurance levels, which can potentially be calculated through all the different controls that you're doing on a system. The kind of one of the most guaranteed ways of coming out with a sterile product at the end of your process is terminal sterilization. But not every product has a way to be terminally sterilized. There are things that cannot be, for instance, heated for too long or treated with radiation, irradiation. So a lot of it is on the front end and on the process itself for producing products. And the microlab really is important there in the quality of data and how they go about demonstrating the facility and processes are under control.
That's a really interesting point that you made about sterility testing, because a lot of, not a lot, but I mean, I think I've heard the misconception before anyways, that, well, we've never had a sterility failure during final product testing. That's great when a site is making, I don't know, say 20, 30,000 units in a batch, right? The number of units that actually get tested for sterility is typically 20 out of 20,000, 20 to 30,000. So you have assurance, 100% assurance that 20 of those vials out of 20,000 are in fact sterile. The other 19,980 vials, you have a high degree of assurance based on the other process controls, your cleaning and disinfection, your environmental monitoring showing that the environment was in a state of control during your batch, personnel, aseptic techniques employed. All that other assurance is provided through risk-based processing and having adequate process controls and the ability to monitor and detect shifts in the level of control of your process. So good point there, Jane.
And because those sample sizes, because those sample sizes are so small, if you do end up detecting contamination and it's valid contamination, what you've most likely detected is gross contamination across the batch.
So those 20 vials that end up being sampled, do those end up going down the chain and like delivered to actual patients, or are those destroyed?
Nope. Those samples, so what that sterility test involves is actually taking the contents of that vial and adding, and there's several different ways to do it, but basically you're adding that product to growth media. So once you've done that, you've opened the vial, right?
Right.
In a controlled environment, so you don't contaminate it during the test, but you've opened the vial, you've then added the product to growth media to detect microbial contamination.
Gotcha.
And so it's a destructive test. It's what is called a destructive test.
Right. So then essentially there's never a test actually directly done on anything that's delivered to a patient, correct?
That's correct. That's
wow.
correct.
Yeah.
Wow.
Yeah.
So it seems like there would be always some nominal risk in that case.
Yes, and that is the statistical approach for verifying that a product has been made aseptically. It's always going to be a numbers game.
Right. Okay, and then there's always just steps that are taken to ensure that those vials that are actually delivered to patient, because patient safety is essentially why we all do what we do. The steps are taken such that the ultimate drug that's delivered is safe.
Yeah,
exactly. So it's a combination of a lot of different controls. Like James was talking about earlier, you have materials. Those materials are tested by QC, before they're released to be used for production, to make sure that they have the identity, strength, that they're pure, and they're not grossly contaminated. You have, people that are involved in aseptic processing are subject to rigorous training in general microbiology, clean room behaviors, aseptic gowning, and it's a really difficult job. It's hard work, but it's working inside of a clean room where you have to have a high level of awareness to everything that you do, because the primary basis for facility controls is airflow, and the ability to work without disrupting that airflow, because that airflow pushes everything down. So if there's contamination in the air, it's being pushed down and then swept away through the air returns that are low to the ground. So if you disrupt that airflow and create eddies in the air, that can swoop down and pick up contamination and bring it to working level.
And that's the dream.
Yeah.
That airflow that you were talking about, and that is not the case in all facilities. Ideally you have supply up high and you've got returns down low. I think we've all seen facilities where there maybe aren't a lot of returns on the ground, and maybe there might be some up high. So you don't know what is going on with the airflow.
So, yep. And so you have, you test the raw materials, you train the personnel, you have facility controls that are engineering controls. Like I was saying, the airflow, you have robust air change rates to make sure the air is constantly being refreshed and replaced with clean, fresh air, HEPA filtered air. And then you have product testing. So a sample size of your overall batch is tested for sterility. It's also tested for different product characteristics, strength, identity, purity, sterility. And so all those things combined, testing of materials, facility controls, training personnel, all of that has to be, goes into consideration when you release a batch for the market, so that patients can, so that the drug can be administered to patients. And so QA in the United States, QA is responsible for releasing those batches. And so they have to look at that during batch release. They have to look at the environmental monitoring. They have to look at the facility controls, the personnel interventions that were documented, and make sure there's a high degree of assurance before that batch is released. It's a very big responsibility. In the European markets, they have what is called the qualified person, and where in US the responsibility lies with the quality assurance unit. In Europe, that responsibility lies with the qualified person. And so the difference being all the responsibility lies with one person in Europe, whereas the responsibility is shared by the, quality assurance unit and, the person that is the head of the site that releases the batches,
Well, that's a really important, like, sort of regional difference that you've highlighted. So what do you think is probably the most, I mean, I guess there's probably positives and negatives to either approach. So what are some of the downsides of having a team-based responsibility rather than a single qualified person?
That's a good question. I think it comes down to accountability.
Mm-hmm.
In the EU, the qualified person, the accountability goes all the accountability goes to the qualified person, right?
Mm-hmm.
And so that mitigates risk, I think, in some way, because the qualified person then has to make sure that the source of the materials, like James was talking about, source of materials is qualified, that sites have adequate training, adequate facility design, adequate facility controls in place. And so they have to audit, and and do those checks before they release a batch. Whereas, in the United States, a lot of responsibility lies with the quality director. They're typically the ones that are, releasing batches or responsible for the quality unit, which releases the batches. And so the accountability lies with, the site head, the quality director, but it's also pushed down to the rest of the quality unit to ensure that there's a thorough review of all the batches. And the QP also relies somewhat on the quality unit as well. But the responsibility ultimately lies with the QP if something goes wrong with that batch.
Mm-hmm.
Whereas, it's more of a site responsibility or a site function in the United States.
And so the QP or the not QP kind of discussion is, visually, I'm kind of perceiving that as like what are your responsibility eggs? Like, are they all in one basket where there's one person responsible?
Mm-hmm.
And hopefully that basket is well organized and they know where all the responsibility that they have to hold, percolates up through the organization. But not having necessarily a basket where your site head has all the eggs laid out. They have a whole lot of, and they may not be, necessarily from a quality background, your site heads. So, so they would have to depend on an assortment of, directors, supervisors, where that quality responsibility is distributed. I mean, there can be positives and negatives for either of those situations. Because what if you lose your QP? What if the one person that has it all, has a whole world, quality world for a system in their head? What if they move on?
You have to find another QP.
Yeah.
You got to think about succession planning as well.